this lecture corresponds to chapter 9, right? So we just finished chapter 10, photosynthesis. We're going back now and doing chapter 9. And if you go through the book linearly, this chapter is coming hot off of the heels of the energy chapter. So they're going to talk about some of the concepts there that we've already covered. So it's a little backwards the way we're telling the story here in class. And let's start off with a little joke here. In case of loss of cabin pressure, oxygen mass will drop down automatically. And you see the fern sitting there going, ah, great. So if you don't get this joke, you haven't studied. This is a joke quiz, right? So the idea there is, the reason why the fern's not happy is because they don't breathe oxygen, right? They give off the oxygen. Anyway, I've already given you this one. I'll skip that one. And then here's the beginning of today. Like, one does not simply break down glucose. It is not a simple process by any stretch of the imagination. If you don't get that reference, you haven't watched Lord of the Rings. And really, this meme is like 10 years old now. Actually, it's 10 years old now. I rewatched this movie recently and I realized it came out 10 years ago. And I felt so old. I still remember opening night. Anyway, how about this one? Once I told a chemistry joke, there was no reaction. Yeah, I got some smirks. That's good enough. All right. And what if I told you the citric acid cycle happens in the matrix? That's where it happens. Remember the mitochondria? The inside of the mitochondria is called the matrix. And that's where it happens. Anyway, all right. So we're not going to go through this. This is what we're going to do today, or at least we're going to get through about half of this today, and then we're going to finish it on Wednesday. And just as a perspective here, the average human adult, requires about 2,200 kilocalories of energy a day just to maintain the status quo. And about 3/4 of that energy goes to just being. You can be in bed all day and you're still going to burn 75% of that energy. 25% gets used for physical activity, right? Just think about that for a second, right? So one of the most common fallacies of people who think they have to go on a diet to lose weight, or rather that they have to exercise a lot to lose weight. It turns out exercise doesn't contribute so much to weight loss as opposed to diet. Diet is everything. Exercise is essential for this, for our brain's health. And it's good for our immune system and getting our lymphatic systems moving and stuff. But purely from the perspective of losing weight, as Americans who are sedentary and we eat terribly high caloric foods, the strategy needs to focus more on the dietary aspects and less on the exercise aspects, it turns out. In other words, you can do all the exercise in the world, but if your diet sucks, you're still going to be overweight. And there's a whole bunch of psychology there too. As you exercise more, your metabolism goes up, you get hungrier. And so we've all, for those of us who exercise, we all feel like, oh, I just ran 5 miles. I'm going to reward myself and eat all this pizza. Okay, right? I mean, I've been there myself many times, right? The trick is just don't eat so much, Andrew, and you won't have your boombalati. As like my friend Scott called it that. He called it his boombalati. And so the boombalati is due to too much pizza, basically. I saw that, Julie. All right. So what's a kilocalorie? We've already talked about this, but here's the slide on it. A kilocalorie is 1,000 calories. What's a calorie? A calorie is the amount of energy it takes to heat 1 gram of pure water up by 1 degree Celsius. Right? Liquid water. And the reason why we have calories versus kilocalories, right? So there's capital C calorie versus lowercase calorie. The capital C calorie we call food calorie. And this is just to convert. It's the same reason why we don't measure car efficiency in inches per gallon. right? I mean, how fuel efficient is your car? Oh, I get 200 billion inches per gallon. That you mean 30 miles per gallon? That makes more sense, right? It's just our brains and comprehending things. So we measure the food calories as thousands of calories because everything has hundreds of thousands of calories. So it's just scaling it up. And instead of saying kilocalorie, because apparently the American public isn't equipped to handle the metric system in any way, we just call it a calorie again. But we just make it a capital C.
It's funny
Yeah.
if you ever like tell people who are like complete health nuts that, and then they're like, that's how many calories you eat. It's like, yeah, like that's how much you need. Like, it's just my one friend, like he's like always counting his calories, like complete like nut job. And like I told him that he like lost his mind. It's funny.
Yeah, most people don't realize that your brain needs a tremendous amount of energy to do its everyday work. And so if you're starving yourself, you're actually going to, if you're particularly starving yourself, you're going to suffer mental consequences. It is important to realize that the more studies they're doing, remember, science is replaced by better science, right? So the current science very suggests that when you go into starvation mode, when you live in a calorie restricted lifestyle, that seems, at least in mouse models, to greatly extend longevity. So mice on diets that are calorie restricted live much longer than mice that are not calorie restricted, which is really interesting. So I guess feeding all these blueberries to the vole is just speeding up the inevitable demise. How are you doing there? You still there? Yep, there he is. I put a tea towel over him so I don't see him during the day because I figured I'm going to get distracted otherwise. He lives right next to me now.
I was wondering why you put that on there. I thought there was a good reason.
Yeah, it's because of my own ADD. If I saw him just constantly moving around, I would be able to teach you guys. But also, I think they're burrowing mammals. So I have a feeling that by keeping that on there, he feels like he's underground and safe. Whereas I put him in the sink, he's terrified. When I clean out his cage, I go and put him in the sink, and he does his cocked head eye thing. He's looking up at me with just wise, like, runs around. And I think he's looking for hawks and owls because everything eats them, and so they're just terrified out in the open, right? It's like being in a war zone while bullets going everywhere, as far as he's concerned. So I think he's feeling less anxious, and I'm not being distracted until just now.
Are you gonna take him to the vet?
Well, again, I caught him. He's probably at the end of his life to begin with. I'm just it's like a hospice situation, I think, because again, their lifespan is 3 months and he's an adult. So he's probably at least 1 to 2 months old. So he's already a geriatric patient, so to speak. Although he's getting better. And the other thing is, I looked this up. You can't do anything for the toxoplasmosis gondii or whatever it's called. They're not going to do anything for a vole. I'm going to hand it to them and they're going to say thank you, and then they'll just anesthetize him. I mean, it's a vole. Or they'll feed him to a snake. These are not high priority animals. They're not endangered. They're pest creatures. If you have a garden at home, these would be things you hate, because they eat the roots of any succulent plant or any little baby plant. They will just destroy your garden. And they multiply really, really quickly. So, yeah. So no, I'm not taking him to a vet, but he's okay. He just runs in circles. Anyway, all right. Good calories. Here's the next level up for you guys. When we talk about respiration, we normally think about breathing in and out, right? We talk about us respiring. We breathe in oxygen and out carbon dioxide. You can now classify that as organismal level respiration, right? So that's the idea of your lungs and your cells and the cycles of gas exchange here on the left. What we're talking about today, when we say cellular respiration, we're talking about what goes on inside the cell itself. And so what's important to remember is that cell respiration can be aerobic, meaning it requires oxygen, but there are also anaerobic respiration mechanisms. We don't do these. Humans are not able to do anaerobic respiration. We do fermentation when we have to. there are prokaryotes that undergo what's called anaerobic respiration. So they have electron transport genes. The key is there is another final electron acceptor besides oxygen. And we'll talk about that at the end of the semester, or at the end of the lecture. So what cellular respiration is, this idea of taking some organic compound that has free energy in it. That's the key here. It has to have some free energy. And you can think of it like a combustion. In the presence of oxygen, we're gonna oxidize it. And we're gonna break it down into carbon dioxide and water, and we're gonna pull that energy out in the process. So think of it as a series of redox reactions. Now for sugar, for glucose in particular, the number you might want to burn into your head is the release of free energy from a single glucose molecule is equal to 686 kilocalories per mole of glucose. So it's a delta G of negative 686. It's negative because glucose is losing that energy, right? And so here it is in a couple of different ways. Your book and some other books, but the idea is the same. We're going to take a high energy molecule and we're going to oxidize it to low energy molecules. And what we're going to do is we're going to use glucose as an example. So you can do this to amino acids, you can do this to fatty acids, you can do this to phospholipids in theory. Okay, the only thing we don't generally use as a fuel source are nucleic acids because they're toxic. They have all that nitrogen in there. So we don't eat DNA and RNA. I mean, we do, they're in all our foods, but we don't extract the energy from them. We have to deal with them. In contrast, the proteins, the lipids, and the sugars, we extract energy out of all of those, right? And there is a pecking order. And the pecking order is from sugars to fats or lipids to proteins. Your body, the mammalian anatomy prefers it in this order. Okay, so everyday life is all about sugar in the blood. When we go into starvation mode, we start tapping into our fat reserves. If there are no fat reserves, we start cutting into our muscle. So a human that is starving, somebody who has gone weeks and weeks and weeks with malnutrition and doesn't get enough calories routinely, they will be literal skin and bones. They will have just muscle, no fat. And then what happens is they start getting a gas buildup in their stomach. And that's why they have that inflamed stomach. That's gas. It's not fat. That's when you know you're on the last legs of approaching a lethal situation. You can go about a month without eating food. You can only go a few days without water. Although if you do go a month without food, you'll probably suffer permanent neurological damage and liver damage. And there's all sorts of really, really nasty things that happens in the process of long-term starvation.
Isn't it like if you don't eat, like I'm pretty sure I learned this with like the Holocaust, and when they started to try to eat again, like you can't, or like your body's not used to it, so you have to work yourself up to it again?
Yeah. If you
Your
take
stomach
somebody,
hurts.
that is true. If you take somebody
Physically.
who's starving for a long period of time and you give them like a Big Mac, they'll just vomit it right up. They can't. They're because the enzymes are all shut off. Their bodies are conserving their energy, right? So because they've gone so long without breaking anything down. Remember the lac operon? I covered that in my micro class. There's this idea of controlling gene regulation, right? So when there's nothing there to break down, it would waste energy to make the enzymes to break them down. So we stop making the enzymes. Yeah, so you gear it up slowly over a period of days. Water first, simple sugars. Now, I'm by no means an expert in any of this, but that's
Your
the
hunger
gist of it.
cues turn off too.
Say again?
Your hunger cues,
But
like
what about?
in your brain, like telling you like I'm hungry.
Oh yeah.
Your hunger cues, they turn off, like because it's a waste of energy since you're not listening to it. Your body like won't use that.
Yeah. Actually, I learned about when I was teaching the anatomy class last semester, I learned about your primary and secondary insulin response. So insulin is a hormone that takes sugars out of your blood and puts them into your liver. That's how we, so when you eat a lot, the insulin takes that sugar and stores it for you, right? That's a good thing. And so people who are diabetic, they're type 1, they don't make insulin, or if they're type 2, they're basically so addicted to insulin that they're not responsive to the normal amounts of it anymore. So, but when you smell something good, right? We've all had that, like we salivate, and it turns out we also have an insulin response. We kick in our insulin levels. And so one of the issues with diet soda, and this is something I've learned empirically, I used to be addicted to Diet Coke or Diet Pepsi. I noticed that diet sodas would make me sleepy. And I think what was happening was the sweetness of it would trigger my primary insulin response. And so insulin would start getting kicked on, but then there's no sugars that are flooding my blood because it's a diet soda. And so the insulin would take what sugars are in the blood and pull them out. And then I would get sleepy, almost like a sugar crash. So I stopped drinking so much soda. And when I do drink soda, I drink normal soda, which isn't healthy either, but everybody's got their thing, right? But I do notice that the lethargy part went away. Anyway, yeah, so if you like this stuff, this is all the realm of physiology. If you want to, dig into that from an academic perspective, you take AMP1 and AMP2, and you'll learn all this stuff. And then nutrition. There's a nutrition class you can take too. All right, so here's the 60,000-foot version of this. We're going to take glucose and oxygen, and we're going to break them down into carbon dioxide and water. And notice that there's multiple paths. Do you see the multiple arrows? It's not one arrow. There's multiple arrows. And then on the other side, right, we're going to couple this breakdown of sugar to the generation of ATP, which is the middleman to go over to the various work of the anabolism to build everything we need. Right? So this is us eating our foods to building everything we want. And so collectively, we talk about our metabolism, right? And it's important to realize, again, plants do this too. They just make their own glucose. They're not getting it from external. So the whole point is where are you getting this? That's what makes you the autotroph or the heterotroph, right? Autotrophs make their own sugar. Heterotrophs eat somebody else's sugar. Thank you for your service. Nom nom. I love this slide. It's one of my favorite slides. So why are there so many steps to the process, right? Why is this so damn complicated? The reason is very simple. If you have an energy reserve, like a gas tank or a chicken wing or a chocolate chip cookie, if you take all of the energy within that reserve and release it all at once, think of the second law of thermodynamics, right? Think back when we talked about that pool ball example, right? If you have a reserve of energy in the pool ball, when you hit another ball to go forward, remember all the secondary consequences of that, right? You hear the clack sound. You're heating up the carpet of the pool table with some heat. You're warming things up a little bit. You're pushing the air out of the way, right? There's all these things that also happen besides what you want. So for the case of a car, what you want is for the car to drive forward in a linear motion, right? If you take all the gas in the car at once and combust it all at once instead of in little bits, you get the car moves maybe a few feet in one direction, and all that energy gets used doing other things like braking the car and vastly heating up the environment. Okay. As for the Mr. Bean chicken wing example here, I use the chicken wing as a really good example for this. So I want you to imagine you go out to eat with your friends pre-pandemic, or maybe next year, whenever you're able to go out with your friends again. And you already had a big lunch, but you're doing a social thing. So you go out to dinner and somebody orders a big thing of wings, and you love wings. Just pretend if you don't love wings, you love wings in this example. But you're not really that hungry, but you take a wing anyway, So what do? You probably take a bite or two of it, and that's that, right? So I want you to think about what's left over. That wing that you took a few bites out of has plenty of energy left in it, right? Somebody else could eat that. Now let's imagine that you haven't eaten in weeks. You're lost in the woods and you just didn't eat anything. You didn't forage for berries or anything. You were just too stressed, right? So you get out of the woods, you get rescued, and they put you in the nearest diner and they stick a single chicken wing in front of you, crispy and hot, with a little side of blue cheese. And of course, you start salivating. You haven't eaten in weeks, right? And you can imagine how you're going to eat that chicken wing. Even if you don't like chicken wings, you're going to eat the chicken wing. Even if it's like diametrically opposed to your philosophies on life. You're starving and this is food, right? So you're going to eat all of the skin, all the bone, not all the bones. You're going to eat all the meat off the bones. And you're going to get to the point where you are going to eat all the little weird ligament stuff that usually nobody pays attention to. And you're going to get to the point where you have that bone, you're going to crack it in half, and you're going to suck the marrow out of the bones. No, you've never been starving. You don't know. No, neither have I. But the idea is there's untapped energy reserves. It's harder and harder to get to it, right? That first bite's the easiest. You have a few bites, and then again, if you're in a land of plenty, and you have 1,000 wings and you're not that hungry, you might take a bite or two and throw them out because you just like the good parts, right? But let's say you only have 3 wings because you're sharing. Now you're going to eat, you're going to take time to get to all that little harder to get meat, right? And if you're truly starving, you're going to start to access this stuff deep. That's hard to get to. And so what we're going to see here is there's a quick burn. Glycolysis is easy and fast. Everybody does it. The products of that are like a chicken bone. Okay. For bacteria that don't go through cell respiration, for bacteria that just do glycolysis, that's the end of their story. They don't get a lot of energy out of the original glucose molecule. They only get like 2% of it. In the presence of oxygen, we're going to tap into that bone marrow and we're going to see that that's where the good stuff is, it turns out. Right? And that's the end of the analogy. Bone marrow doesn't have any particular great energy reserve in real life. It's not like you should really, I'm not encouraging you to eat bone marrow, but that's the analogy here. That's what the untapped potential is for glucose's energy. We're going to see that in the presence of oxygen, when we take that byproduct, pyruvate, we're going to be able to get up to 30% of the energy out instead of a measly 2. That translates to, instead of 2 ATP, 30 ATP. Okay. And so the idea here is if we're inefficient, if there are metabolic process, so the reason why metabolic process have multiple steps is to maximize the efficiency of the energy extraction. In the same way our car is fuel efficient. We just, with that little buzz of the spark plug blows up just a little bit of the gas in your tank at a time that's in the cylinder of your engine. And that is what allows us to drive forward. And instead of getting a foot out of 30 gallons, we can get 300 miles out of it or more. Okay. And of course, Cookie Monster is the least efficient of these. Why? Because he's a Muppet. They don't actually eat anything. Zero. all that food's going to waste. Every cookie that woman's putting in front of him, 100% of the energy is going to waste. All right. Another reason to think about is that you can't really do anything useful with a large energy reserve. Right? Imagine going to CVS and all you want is like a bottle of water and a pack of gum. It costs a total of like $4.75, and you give them $1,000 bill. They're just gonna look at you like, I can't. Or how about a brick of gold, right? You can't exchange that.
The
It's not
cashier
useful. Yeah.
right there.
What'd you say?
So the cashier probably would have quit right then and there.
Right. They're just like, I don't know what to do with this. This is more than I make in a month. Yeah, I mean, some people, that's why we're all going to school, right? Get higher paying salaries. So the idea here is glucose itself, one molecule of glucose, 686 kilocalories. Think of that like $1,000 bill versus a molecule of ATP, 7 kilocalories, 7.3. Okay, this becomes the useful energy exchanger, right? It has enough free energy in it to get common tasks done. And if you need more of it, you know, what you have dollar bills, right? Oh, here, 1, 2, 3. I need 7 ATP to do this process. I need 2 ATP to do that process. That's why we use ATP. It's an efficiency issue. Now, the key player for cell respiration we're going to see is the mitochondria. So it pays to know a little bit about the mitochondria. So go ahead and review your anatomy back from chapter 6. That's your cell chapter. Inner membrane, outer membrane, the matrix, the space, all that. Remember, there's a chromosome in there that's producing its own dedicated ribosomes and all that. They divide by binary fission. So the idea is they used to be very long ago free living microbes, prokaryotic cells that are now part of our own metabolism, right? But profoundly, I'm going to go to the blackboard here for a second. If this is your cell, this is the mitochondria. Okay. If you remember, the idea here of endosymbiotic theory. So pre-endosymbiotic theory, we were still living, we had cells, right? And so glycolysis, the idea here, that's always been happening in the cytoplasm. And so that's where glycolysis occurs, cytoplasm, nothing's changed. All the fancy stuff's happening in here. This is where we're going to need the oxygen. And so technically your cells are still anaerobic. The only place we're going to use oxygen is in the heart of the mitochondria. Right? So glycolysis turns out to be one of the, if not the most ancient metabolic processes on the planet. It's still going on, 2.5 billion years later after it evolved, and it's never stopped. It's one of the most continuously running metabolic processes on the planet. Every cell type on Earth uses glycolysis. Archaeans and bacteria and fungus and protists and plants and animals. Everybody, all walks of life can use glycolysis routinely. We've already covered the key players of electron carriers. We've talked about this before, so I'm just going to very briefly review this idea. We need a way of taking high energy electrons and physically transporting them into the other parts of the cell. And so that's what these dinucleotides' job is. Unlike plants that use NADP, the phosphorylated version of NAD, the mitochondria and everything else that we're talking about today is just NAD, and then we'll see in the Krebs cycle there's FADH. And really, you could just reduce that to FAD2. You got NAD and FAD. Nicotinamide adenosine dinucleotide. I'm sorry, adenine. This is one thing I frequently make a mistake about. That's adenine. It's nicotinamide adenine dinucleotide, not adenosine, not dinucleotide. The smaller of the words. And this is flavin adenine dinucleotide. So you got nicotinamide and flavin are the two new ones. But these are, at the end of the day, they're just two nucleotides stuck together. Okay. All right, now what we're gonna do here, I'm gonna whiteboard the beginnings of this. And then on Wednesday, we'll do the PowerPoint for this. So I wanna give you an overview here. So first, let's talk about the goal of it all. We're going to have this red hot molecule, glucose. 1, 2, 3, 4, 5, 6. All right, let's do it linearly. Let's do it this way. I said 1, 2, 3, 4, 5, 6. Here's glucose. That's what we're going to start with, right? So where do you come from? Remember photosynthesis. We had 2 molecules of G3P, stick them together. There's our glucose. So glucose gets into the cell. Here's the cell membrane. Glucose gets into the cell through a transporter. It's called a glucose transporter. And in our cells, that's mediated by insulin. Insulin helps glucose get into cells. There's also an enzyme produced by our thyroid, thyroxine, that helps regulate the speed of this. This is what helps keep our basal metabolic rate in sync. All right, so we're going to take glucose, we're going to stick it inside the cell. We don't want glucose to leave. All right, you know what? Let's stick with the 30,000 foot view first. What we're going to do here is we're going to see an enzymatic series of reactions, 10 reactions to be precise, that are going to culminate in a splitting of the glucose and basically see the Calvin cycle running in reverse here, if you look at the detailed mechanisms of it. And we're going to get 2 3-carbon molecules that are still pretty high in energy. This is pyruvate. So this is a downhill reaction. This is spontaneous. This is exergonic. All that good stuff we talked about in the basics of metabolism. And in the process here, we're going to need to put in 2 ATP to get the ball rolling. We're going to get out of it 4 ATP. And so we're going to say we net 2 ATP. We use 2 to get 4. It's a good investment, right? In addition, we're going to take, here, think of this as an empty electron carrier. Think of it like an empty bucket. And we're going to take 2 empty buckets. And we're going to fill them filled with energy. There. Oh boy. Let's try this again. This bucket, of course, is NAD. NAD plus, NADH. And the reason why I want to show you that bucket, and the reason why I took so much trouble to draw that, I want you to really get the idea that we need that empty bucket to do this reaction. If the buckets are full, we can't keep going. So we're going to see at some point, we're going to have to dump those buckets to come back here to keep this process going. Does that make sense? From a conceptual standpoint, right? So this is required. In the absence of that, we can't do this job. Okay, we need a place to put the energy. If we don't have a place to put the energy, we don't do this. Okay, so this is glycolysis. Glyco, sugar, lysis, break in sugar. That's what I just mapped out in a very simple way. That's the concept of glycolysis that occurs in the cytoplasm. It is an anaerobic process. In other words, it does not require oxygen. This just happens. Okay. And so, by the way, yeah, there's 2 of these and there's 2 of these. For each 1 molecule of glucose, we will produce 2 molecules of NADH and 2 net molecules of ATP. And so think of the products now. There's 3 products. I'll just highlight them. There are the 2 pyruvates. Don't forget that. There's the 2 pyruvates, there's the 2 ATPs, and there's the 2 NADHs. Those are the products of glycolysis. And yes, there's some water in there. Don't worry about the water. Water just gets made everywhere. Okay, you still with me? Still making sense?
I have a question.
Go ahead.
Are you talking like, I guess just generally like NAD plus is going in and then NADH is going out, or is there like a, I guess, a specific step in which?
Oh yeah. There's
Reduce.
2 specific steps that happens. Actually, if you look at photosynthesis, Calvin cycle, where we made the NAD, I'm sorry, remember we used the NADH to make G3P. It's the same chemical reactions in reverse. Those are the same 2 steps. All right, so ready? I'm going to now redo this in a summarized way here. Let's try this again here. So 1, 2, 3, 4, 5, 6. Glucose. All right, here we go. So the first few chemical reactions we collectively call the energy investment phase, or just energy investment, right? So that would be phase 1 of glycolysis. It is a series of 3 chemical reactions that you don't have to know the details of. Okay. And again, don't worry about the names of the intermediates right now. That's phase 1. And so that's going to use up 2 ATP. So we're spending money right now. All right. So there's glucose. Don't worry about the names. The second, you know what? I'm drawing this. I'll tell you what. And my apologies. I want to make this clearer. I want to make this as clear as possible. I didn't make any, there's no mistakes there, but I'm going to do this. I'm going to exaggerate that first arrow before we break anything. 1, 2, 3, 4, 5, 6. So think of it here. Here's some phosphates. And then, so that's step 1. That's the phase 1, 3 reactions there. Then the second step is what we call the cleavage phase. That's where we break it in half. Cleft and twine. And of course, there's some phosphates here. And then the third step is the payoff. We call this the energy. Well, different books call it different things. Let's call it the energy liberation phase. And what we're going to do here in another series of, I think those are 3 reactions. This is when we're going to take our NAD. There's 2 of them. And we're going to pick up that energy here. We're also going to make ATP in a way we've never made it before. The last time you've seen making ATP, we used a proton gradient and ATP synthase and cranked it out. What we're going to do here is we're going to use this molecule as a substrate to phosphorylate. And so this is called substrate level phosphorylation. We're going to take ADP and make ATP, and we're going to make 4 of those in this process here. We're also going to use some inorganic phosphates, but we're going to basically rip those phosphates off. So that's a source of phosphate. And so we call this substrate level phosphorylation. Okay, so this is a unique way to make ATP that you haven't seen yet through a metabolic pathway, basically, instead of a proton gradient. Again, recall, remember the thylakoid, we fill up the protons, and then we have the ATP synthase, and these flow downhill, and we make ATP. This is different than that. That was the only way you've made ATP before. We're not doing it that way. We're doing it instead using enzymes and substrates. So it's called substrate level phosphorylation. And we don't make that much of it. We're making 4, we use 2. So we say we net 2, right? We used 2 in the first phase above. we made 4 for a total of 2. Okay, and so the final product here of the energy liberation phase, also the penultimate product is PEP. You've seen that before, remember C4 plants. And then finally, we make that last phosphorylation event and we get pyruvate. You can also call that pyruvic acid. It's in solution, it's an acid, so it'll dissociate. Okay, so that's glycolysis. That all occurs in the cytoplasm. Okay, so to summarize the products there, you got your 2 pyruvates, your net 2 ATPs, and your 2 NADHs.
Excuse me?
You're excused. What's your question?
Okay, so the 4 ADP that are input and then they're phosphorylated to ATP. So the energy that is needed to do that, is that coupled when the NAD turns in NADH?
No, the energy to create the ATPs comes from the free energy within the G3P molecule itself and the BPG. So you have G3P, and then you have BPG, that's bisphosphoglycerate, and then you have the phosphoglyceraldehyde, or the 3PG, right? So the energy, those are high energy to low energy molecules. And so we're giving up, each one of those conversions is an exergonic reaction, and we capture some of that energy in the ATP.
So like
And
that's
then,
all.
say again?
Sorry, I was gonna say, so like that's all happening, like that breakdown is happening. It's releasing energy, which is like forcing the phosphorylation then?
Yeah, you got it. Yeah, so there's enzymes. The enzymes take that free energy and staple the phosphate group onto the ADP. You got it. And again, all of this is happening in the absence of oxygen. Any other questions? Okay. Let's now look. So I'm going to just summarize that again. I'm just going to doodle the carbon. So we're going to go to the next step here. 1, 2, 3, 4, 5, 6. One, two, three, 1, 2, 3. All right. So here we are in the cytoplasm. Now over here, there's the mitochondrial membrane. And here's the inner membrane and the outer membrane. Let's think about oxygen now. This is where we fork into 2 parallel universes here. We're either going to do something without oxygen, we're going to do something with oxygen. Okay. In the presence of oxygen plus O2, we're going to see the pyruvates get pumped into the mitochondrial matrix. In the presence of oxygen. We're also going to see, do you remember the NADPH that we made? I'm sorry, NADH. The 2 of them. Again, in the presence of oxygen, we're going to see, don't worry about any of the details here. And this is going to come into the matrix now, because that's where we're going to use it later. And it has 2 possible fates. Either when everything is said and done, it remains as NADH. Or in some overly complicated process we're not going to learn about, it ends up as FeDH2. So the electrons find themselves in the matrix one way or another from the cytoplasm. They're useless out in the cytoplasm. There's nothing we can do with them in the cytoplasm useful, because we're going to see, I want you to recall now, this is why I teach photosynthesis first. I want you to think of photosystem 2, grabbing the sunlight, pumping that electrons full of energy, and then handing those energies off, or handing those electrons off to the electron transport chain, right? High energy electrons being handed to a chain. These things do the same thing that the photosystem does. It hands those high energy electrons off to a chain. Okay, and so that chain is in the matrix. It's really it's in the crystal membrane here. But we're not focusing on that detail just yet. I just want you to show in your notes and in your brains right now, we're going to put those things to work. The only place they can go to useful work is here in the matrix. And you can only get there in the presence of oxygen. In the absence of oxygen, instead what happens is we're just going to dump that energy. We're going to waste it by dumping it enzymatically, of course, onto this pyruvate so that we can regenerate that NAD. That's what fermentation is. So fermentation, think of fermentation as an extension of glycolysis that only occurs in the absence of oxygen, and its goal is to regenerate that free NAD so that it can keep glycolysis going. It empties the bucket. And the products of that depend, I would say products depend on the genetics. Depends on which enzymes are present. So we're going to learn about lactic acid fermentation or ethanol fermentation. You don't have to know the details just yet. And that all happens in the absence of oxygen. And again, there's no new energy formed. There's no more efficiencies. There's no additional ATPs. All it does is it frees up that NADH to go and continue the work of glycolysis so that it's a sustainable cycle. So in other words, that NADH is wasted. It's a waste of energy. It's a chicken wing with one bite taken out of it that you're throwing away. It's horribly inefficient. Fermentation is an inefficient process, but it keeps the cell alive. Now let's pick up that story with the presence of oxygen. Let's go into the mitochondria here. The first thing we're going to do to these 3 carbon molecules is we're going to get them ready for their final death in the Krebs cycle. We're going to prep them. This is literally called the preparatory reaction. Your book does not list this as a formal step in the process. I do in my notes, in my lectures, in my PowerPoints. This is formally my step 2, but it's just but it's a simple enough thing where your book just kind of sees it as a middle layer between glycolysis and the Krebs cycle. So it's semantics. You don't have to worry about it. The thing here is we're going to oxidize this pyruvate. So you can also think of this step as pyruvate oxidation. And the enzyme's name appropriately is called pyruvate dehydrogenase. And your book teaches you this, and I'll say it out loud now. A dehydrogenase enzyme is an enzyme that oxidizes things. So whenever you see the word dehydrogenase, you're thinking redox. And what we're going to do is we're going to pop off one of those carbons. So here we have a 2 carbon molecule now and a free carbon. And that free carbon, that's carbon dioxide. And of course, this happens twice, right? We have 2 of these molecules. And these are a gas, and these diffuse away, and we breathe them out. That's the carbon dioxide you're exhaling right now. This is one of the places that we generate the carbon dioxide. And I want you to consider the carbon accounting now. We have this initial 6 carbon molecule that broke down into 2 3-carbon molecules. And now we have liberated 2 of the original 6. We still have 4 of the original 6 to deal with. These turn out to be, that's vinegar. That's acetic acid. And of course, if that's not the end of the story with these, we're going to take those are now the chicken bones with the bone marrow trapped inside. This is the last vestiges of energy we're going to grab. And so what we're going to do is we're going to attach these molecules to a coenzyme with the most boring name on the planet. It's called coenzyme A. That's CoA. And so this collectively, this molecule here with an acetic acid attached to a coenzyme, that acetic acid is now technically a functional group. And so we can call this acetyl CoA. And that's what this molecule is. So we make 2 molecules of acetyl CoA here. 2 pyruvates came in, 2 acetyl CoAs leave. And in the liberation of that energy, that dehydrogenase is able to manufacture for each one of those a total of 2 NADHs. So we've trapped more of the energy of glucose now, right? So the product here, we got 2 NADHs, we got 2 acetyl CoAs, and 2 carbon dioxides. What came in were the 2 pyruvates. Okay, so this 3-carbon molecule has a carbon popped off, and in that process releases energy, and that energy is put into an NADH molecule. And the resulting product of that is stapled to a coenzyme. Questions? Go ahead, Alexa.
Where did the NAD plus come from, and where did like the electrons that reduced it come from?
Okay, so NAD plus is everywhere in the cell. There's not a lot of it. It's an expensive molecule, but I want you to think of it as just floating around. And that's a great question, because that's something most of us don't really think about. Where are all these things coming from? You can think of the cell as filled with this very rich, very lush thing called the metabolic pool. There's all these metabolites floating around. NAD is a metabolite, and it's a rare one though. It's kind of like you're rummaging through your Lego bin looking for the one cool piece that takes forever to find. Okay, that's why out in the cytoplasm we needed fermentation. There's not a lot of this stuff. Okay. Oh, that's a great question, Tyler. How fast do these processes occur? Billions of times a second. Like crazy fast. So fast that, yeah, things happen at the molecular level that we would have to slow things down. We couldn't film this, let's put it that way. Instead, what we do to study these things is we use lots of it. That's one way to do it. And then we can also inhibit processes and then watch what builds up as a result of the inhibition. Yeah, so we're coming to the table here after decades of other people doing all this work to pull all this apart. This was insanely complicated to figure out, right? So yeah, it's happening. That pyruvate dehydrogenase enzyme can process millions of molecules a second. And it's actually really, if you look at the PowerPoint, there's this amazing picture of it. It's a sphere within a sphere that rotates inside. And so you can think of a whole bunch of like a swarm of bees coming in on one side and leaving on the other side as CoA, just all at once. All right, more questions. Oh, so wait, Alexa, the second part of your question, where are the electrons coming from? Pyruvate. Right, we're ripping pyruvate apart. So we're taking a pair of electrons from pyruvate and depositing them in NADH. And so we're breaking a bond, and then the carbon dioxide gets liberated, and we still have that 2-carbon acetyl group. Right, so the electrons are being ripped out of here. breaking that. So right, these are high energy bonds, right? Pyruvate's a high energy molecule, so they have high energy electrons, and we're going to take that high energy pair of electrons and stick it into NAD. I'm going to grab a proton from the environment and create a bond. You still look confused. Go ahead, ask your follow-up question.
So it's broken into like 4 things then, because like 2 of it's going to CO2, 2 of it's going to COA, and then 2 of it's going to the NAD plus.
Let's see, this is pyruvate. Oh, sorry, so we need CoA here, and then we need NAD. And then the CO2. There. I think that's everything. Now here, I have a slide on this. Let's just have, here we go. Here is the pyruvate oxidation. So you start out with a 3-carbon molecule, you pop off one of the carbons, right? And you deposit that energy into the NAD plus to make the NADH. And then you bring coenzyme A in here and you attach the remaining bits of the molecule to it. Right, so think of this pair of high energy electrons as being deposited here. And the remaining bit here finds its way onto CoA. Okay. And so, there's an enzyme that's mediating all this. That makes sense? Yeah, okay. And again, because we're simplifying things, we're not looking at the detailed mechanisms. There's still going to be some unaccounted for things like where the other electrons come from. Let's not worry about that. Okay, for everybody, the key to this is we now have some useful energy deposited into this bucket. And we still have this relatively high energy molecule, acetyl-CoA, that we can continue to mine for energy. Okay. And that carbon dioxide is a gas that is a byproduct of this. It is a waste product and it goes away. It diffuses downhill out of the cell, small, uncharged. All right. And it's 12:50. I got 10 minutes to get to the end of this. I wanted to do at least one pass. So let's go back here. And again, we are in the mitochondrial matrix and we have just made an acetyl-CoA and some NADH. What we're going to do is we're going to take that acetyl-CoA, we're going to transport it here to the Krebs cycle. And so now I'm going to give you the simple version of the Krebs cycle. This is probably one of the most famous things that all students have to deal with one time or another in their biology career, the Krebs cycle. I'll give it to you in the simplest way I can to start with. So it's a cycle like the Calvin cycle. We're going to end where we started. We're going to start where we end, right? We start with this relatively high energy pair of carbons, right? Covalently linked. That's the acetyl group of the CoA. It's bound to the CoA here. So there's your acetyl-CoA. And let's draw this as a blue. So this is a cold molecule. Oops, too small. 1, 2, 3, 4. Let me think for a minute. Yeah, 4. That is our old friend malate. Remember from the C4 plants? We're going to do a malate though. And here I'm going to put him a little down here. I'm going to give you the end story here. The very last reaction here, the 10th reaction, is to take malate, 1, 2, 3, 4, and oxidize it so that we can have this name here. Ready? Oxalo, OXALO, oxaloacetate. Oxaloacetate is the oxidized form of malate. Malate is the reduced form of oxaloacetate. And that's the very last step of the Krebs cycle. We're not talking about products at all. We're not talking about, like, there's an NADH there. We're not going to worry about any of that, because here's what we're going to do. Here's the first part of the Krebs cycle. We're going to take this 4-carbon molecule and this 2-carbon molecule, and we're going to staple them together in the very first step. And we're going to get, let's make it orange. It's hot, but not super hot. 1, 2, 3, 4, 5, 6. So in other words, the super hot molecule touches the cold molecule and it evenly distributes the heat throughout it, and you get this warm molecule. That's citric acid or citrate. The eponymous citrate, the citric acid cycle. Okay, like pyruvate and pyruvic acid, citrate, citric acid. And then in a series of, again, it's another 10 chemical reactions. We're not going to do the details of. I'm just going to summarize this. Ready? We're going to go through. I'm just going to doodle some circles or some arrows like this. And I just want to get back here. Okay, and in that process, we're going to degrade this 6-carbon molecule to a 1, 2, 3, 4, 5-carbon molecule plus a cold carbon dioxide. And we're going to do that a second time. 1, 2, 3, 4, 5. Pop off another CO2. So there it is. Those are the original 2 carbons that came in, are now liberated as carbon dioxides. Those are the last vestiges of the original glucose. And now we have this relatively warm 4-carbon molecule we're going to further oxidize down to malate. So we go from a 6 to a 5 to a 4. I made that a 5. This is a 4. 1, 2, 3, 4. And we're going to take that warm 4-carbon molecule to a cold 4-carbon molecule. Okay, so now let's talk about, we're getting energy out of this, right? So where's the energy? We're going to get out of this a summary. We're going to get a total out of all of this of 3 NADHs, 1 FADH2, and 1 ATP. In various exit points that we don't have to worry about right now. That's the summary of what comes out. And of course, the byproducts of 2 carbon dioxides. So this mapping here, this is per acetyl-CoA. But remember, there are 2 acetyl-CoAs per original glucose molecule. So the Krebs cycle runs twice per glucose molecule, just like everything else has been happening twice. And so the accounting for a single glucose molecule, 1 glucose molecule yields a total of 6, just doubling all that, 6 NADH, 2 FADH2, 2 ATP, and then of course the 4 CO2. And that 4 CO2 should make sense because that's the original 4 carbons that came in. So one thing you should do in your studying of this is trace the fate of the carbons. Where do they get lost? 6 come in and 6 have to leave. You'll see that 2 of them get lost during the pyruvate oxidation step, and the remaining 4 get lost in the Krebs cycle. Okay. And then bear with me. Here's the last, the swan song of the entire process. We got 4 minutes left. I'm just going to give you the summary of what happens after all this. This is the end game, right? If you ever watched Avengers Endgame, after Tony Stark dies, there's still story that happens. Nobody cares about it because Tony Stark's dead, right? So, but there's still stuff that happens. What'd you say?
I said I beg to differ.
Yeah. So there's all this stuff that happens after glucose is long dead. So let's think about it. We have, remember glycolysis, we had those two NADHs, right? And let's say that, or there are two FEDHs. And then in pyruvate oxidation, we made two NADHs. And then in the Krebs cycle, we made 6 NADHs and 2 FEDH2s. Right, so what I just tracked there was all those electron carriers. Forget the ATPs. We made some ATPs too, but we're just focusing on these electron carriers. Because here we have all of these filled energy buckets with nothing to do with them yet, right? So this right here, this is where we've trapped the vast majority of the glucose's free energy. And so we got to do something with it. And so the very last thing we're going to do is we're going to take these super duper high energy electron carriers, and we're going to dump them into an electron transport chain. So ready? Here's the outer mitochondrial membrane. And here are the cristae. Okay, and I want to draw the cristae simply like this right now, just for this diagram here. And what I'm going to do is I'm going to show you there are, let me draw this really thick. 1, 2, 3, 4. And they're Roman numerals 1, 2, 3, 4. This is the electron transport chain in the mitochondrial inner membrane. This is where your cytochromes are, cytochrome C and all that stuff. This is an electron transport chain. This is a proton pump. And what we're going to do is we're going to pump protons from the matrix into what we call, there's different names for this place. This is usually called the lumen of the mitochondria. You can also call this the intermembrane space. And we're going to fill it with protons, just like the thylakoid space. We're going to increase the concentration of protons, and we're building a gradient, just like the photosystems did, except instead of a photosystem in sunlight donating high energy electrons, we're going to take this NADH and we're going to hand those high energy electrons off to system 1 over here. And then system 2 is going to steal them. Then system 3 is going to steal them. And then system 4 is going to steal them. And at the end of it all is this greedy little golem creature called oxygen that's going to steal the electrons. And this is the whole point of breathing right here. This oxygen and these electrons and the protons everywhere generate water. And so we say here that oxygen is the final electron acceptor. This is what makes us aerobes. That's what makes this aerobic cellular respiration. This chain is sometimes referred to as a respiratory chain. Now, did you see how NADH hands its electrons to number 1? Well, here's what FEDH2. Think of FEDH2 as NADH's little brother. He wants to emulate everything NADH does, but he's not as strong. And so he's going to donate his electrons too, but he can't get them up to 1, so he hands them to 2 instead. Okay, and then here's the final payoff. And here's where we'll end today. Somewhere else on that membrane is our familiar ATP synthase. And just like the one that happens in the mitochondria, I'm sorry, in the thylakoid membrane, this is also going to generate ATP in exactly the same way we learned last time. All right, but here's what I'm going to be able to do for you. We can actually account for how many ATP we make per NADH. And this is where we think of it like a currency exchange. Like how many euros can I make with $1? That kind of idea. So how many ATPs can I make with an NADH? So NADH is handing its electrons off to the pump pumping protons, and then ATP synthase is using that proton pump energy, using the proton gradient to make some ATP. And so for every 1 NADH, you can convert that to 2.5 ATPs. And for every 1 FADH2, you can generate 1.5 ATPs. These are averages. That's why they're decimals. And so because it's 102, I want to cut this short. I'm going to just tell you this right now. When you add everything up, you get about 30 ATP generated per molecule or mole, let's say, of glucose. No, molecule, not mole. 30 ATPs per molecule of glucose. And the reason why we can't be more specific, there's 2 reasons that we'll get to later on Wednesday. I'll tell you quickly now, but if this doesn't make sense, we'll explain it more later. The first is the NADH is from glycolysis. Remember, they can come in as either NADHs or FEDs. So there's a variability there. If they come in as NADHs, there's 2 of them. That gives you 5 ATP, right? 2.5 times 2. Whereas if they come in as FEDH2s, that's only 3 ATPs. So there's a difference of 2. So there's a variability of 2 ATPs depending on what's going on with the glycolysis NADHs. And then the second is the fact that pyruvate needs to get pumped into the matrix, if you remember. The reason, or rather, the mechanism of that pumping is driven by protons. So this proton concentration that we're building up actually does at least 2 jobs for our purposes: make ATP and also pump pyruvate across the membrane. And that pumping is variable. There's not a precise amount of ATP. So it's about 30 at the end of the day. That's the best, most accurate thing we can say. Which turns out to be about 29% of the original 686 kilocalories. When you do the math there, if you do 30 ATP times 7.3 kilocalories per mole, and you get that answer, and you divide 686 by that answer, because that's the total amount of energy in a glucose molecule, you get about 30% or so of the original energy out. So here's my question to you, and this is what we'll end today. Where's the other 70%? What do you think? If I stick a thermometer in your mouth and we read that after a couple of minutes, 70% is wasted as heat. You give off the amount of heat that is equivalent to a 60-watt tungsten light bulb. That's why when there's a bunch of people in a room, it gets hot. Right? Imagine walking into a room with 20 lamps, all traditional 60-watt tungsten light bulbs without any of the hoods on them. They're just burning hot. You'd be like, oh, that's a hot room. That's about as hot it is when you walk into a room with 25 people without circulation. So that's chapter 9 in a nutshell. And we have a whole nother day to do this. Okay, so on Wednesday, we'll go through the slides this time, and I'll give you, the more finesse and more detail. And then time permitting, we'll do a little mini review session connecting the photosynthesis to this. And so come on Wednesday armed with some review questions too on everything from the membrane chapter and the thermodynamic stuff and photosynthesis, right? And then on Friday, we will have our test, our second test. Please don't cram for this. This is the stuff you can't cram for this. You really, I mean, I'm sure you can. It's just, I would not recommend it. You just do a little bit of work, or at this point, do a bunch of work every day. for this stuff. If you can. You have all week, it's just Monday. But all I was saying is like, I want the fire under your feet right now for Friday. Don't delay this until Wednesday, Thursday. You're gonna have a panic attack. Okay. All right. I'll stop the recording.
I have a question.
Oh yeah, I'll keep the recording then. Let's hear your question.
If we have a quiz Wednesday, will it be on both photosynthesis and cellular respiration?
Yes.
Okay.
Good question. Any other questions? All right. I will stop the recording.